Friends, welcome to Biology Medicine. Today in this video, we will discuss about the endoplasmic reticulum. So from the very name you can guess, endoplasmic, that means within the cytoplasm. Reticulum means a net-like. That means within the cytoplasm there is a net-like structure which is known as endoplasmic reticulum. The endoplasmic reticulum is connected with a Golgi body, lysosomes and vacuoles, and all the four together is coming under endomembrane system. Endomembrane system. So this endomembrane system, it includes endoplasmic reticulum, Golgi body, lysosomes and vacuoles. These four are included under the endomembrane system, and they are all interconnected and function in a particular manner. While other organelles like the chloroplast, mitochondria, peroxisomes or other organelles like sperm, they are not part of this endomembrane system. This is important from examination point of view. So the endoplasmic reticulum, Golgi body, lysosome, vacuole, they are coming under endomembrane system, and we will discuss one by one, and this video will discuss particularly endoplasmic reticulum. So from the name, as already I have told you, that the endoplasmic reticulum forms a net-like structure within this cell. Now if we will take suppose this is one cell, this is the cell membrane, and this is the nucleus. The nucleus is a double membrane structure. Now from this nucleus there are some extensions like this, extensions. So these are known as endoplasmic reticulum. Sometimes from the cell membrane also these extensions may come out, cell membrane. So this is the endoplasmic reticulum, and the endoplasmic reticulum was first discovered by Porter, in 1945, and among all the membrane structures that are present in the cytoplasm, it constitutes about 30 to 60% of the membranes is only endoplasmic reticulum. But this endoplasmic reticulum are present only in eukaryotes. They are present only in eukaryotes, and they are absent in prokaryotes, then mature RBC, then eggs, and also they are very poorly developed in meristems or meristemal tissues. Now when this they form certain that is the net-like structure, that means this intracellular space is now divided into two parts. This is which is present within this endoplasmic reticulum. This is known as luminal. This is luminal, while the outer one is this is known as extraluminal space. That means the endoplasmic reticulum divides the intracellular space into luminal. That means within this endoplasmic reticulum, and that which is present outside, that is extraluminal space. Now this endoplasmic reticulum, which is either the extension of the nuclear membrane or the cell membrane, it consists of three different types of structures like cisternae, tubules and vesicles. So usually if we come to its structure, usually it consists of three things. Number one is cisternae. Now usually these cisternae are long, parallel, flat, and sac-like interconnected tubules. That means they will look like this. There are certain parallel sac-like interconnected structures are there, and on the surface there may be ribosomes are present. So this is the cisternae, and usually these cisternae are present. They are usually present in case of these cells which are involved in synthesis. In the cells which are involved in synthesis or synthetic activities like pancreas and brain. So this is the structure cisternae. Now the second one is tubules. Now the tubules are also, you will get, these are tube-like. These are tube-like extensions, and these are usually free from ribosomes. Here ribosomes are present, but these are free from ribosomes, and these are the major structures of smooth endoplasmic reticulum. Major part of smooth endoplasmic reticulum. And how these tubules look like? These are usually, or they may be like this. These are the tubules, and there is no ribosomes on them. Then the third one is vesicles. Vesicles. The vesicles are usually, they are round or oval, and usually they are also free from ribosomes. And they are also part of smooth endoplasmic reticulum. And these vesicles are, they are either round or they may be oval like this. So these are the three, that is cisternae, tubules and vesicles. So these three, they constitute this endoplasmic reticulum. That means in this endoplasmic reticulum, you may get some structures like this, some tubule-like structures or vesicle-like structures. That means all these three, they constitute the endoplasmic reticulum. So this is structure. Now if we'll come to the types of endoplasmic reticulum. The endoplasmic reticulum is mainly of two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. So usually it is of two types. That is, one is smooth endoplasmic reticulum, or in short we can write ACR, and another is RER, or rough endoplasmic reticulum. Usually this smooth endoplasmic reticulum, this is also known as agranular, while this is known as granular. The smooth endoplasmic reticulum, it constitutes about 1/3 of the total endoplasmic reticulum, 1/3 of endoplasmic reticulum, while it constitutes about 2/3 of endoplasmic reticulum. This is one of the differences. Then the second difference is here this is usually the smooth endoplasmic reticulum is usually made up of tubules and vesicles. That means here cisternae are absent, but here these are usually made up of cisternae and tubules. Then there is a third difference. Why it is agranular? Because ribosomes are absent. Ribosomes absent, but here ribosomes present. Ribosomes present. Now the smooth endoplasmic reticulum, they are usually abundant in cells. Abundant in cells like that of the like muscle cells, then adipose tissue, then that is retinal cells. Retinal cells also will get it in case of liver cells, and also the secretory cells. While this rough endoplasmic reticulum, this is abundant in pancreas, then plasma cells, globate cells, and also fibroblasts. This is the basic difference between the smooth endoplasmic and the rough endoplasmic reticulum. It is smooth because ribosomes are absent, and these are rough because ribosomes are present. Now here if ribosomes will be, suppose here ribosomes are present, then this naturally this will be rough endoplasmic reticulum. Here suppose ribosomes are not there, then this is smooth endoplasmic reticulum. Depending upon the presence or absence of the ribosomes, it may be smooth or it may be rough, and both are interconvertible. On smooth, if ribosomes will be there, then it will be converted to rough endoplasmic reticulum, and from rough, if ribosomes will be removed, it will be converted to smooth. So this is about the structure. Now, in case of endoplasmic reticulum, the functions are very important, and while discussing the functions, we will discuss in three different parts. There are some functions which are carried out by both smooth and rough endoplasmic reticulum, while there are specific functions carried out by rough and by smooth. So we will discuss in the three headings: common functions, functions by RER, and functions by SER. So if we discuss the functions, common functions first. That means what are the different functions which are carried out by both smooth and rough endoplasmic reticulum. That is common functions. So among the common functions, the most important is, since it forms a network, reticulated network, that is why it provides mechanical support. It provides mechanical support, or we can tell that it provides a skeletal framework. Skeletal framework. This is one of the important functions. Then the second function that we can there, that is transport within cell. That means it functions as if cells circulatory system. It functions like a cell's circulatory system. Then the third function, that is between this endoplasmic reticulum and the cytoplasm, there will be exchange of substances through diffusion or osmosis. exchange of substances by diffusion and osmosis. The fourth one, since it gives rise to a extensive network, it increases the surface area for different physiological activities. Increases surface area for physiological activities. The next one, in plant cells, two cells are interconnected by means of plasmodesmata, and the plasmodesmata, this endoplasmic reticulum is present in form of desmotubules. So desmotubules in plasmodesmata. Then the next point is localization of organelles. That means which organelle will be placed at which place, and there should not be any link between one another. They will be completely free from one another. That is the function of this localization of organelles. Then formation of nuclear membrane. Formation of nuclear membrane at the end of telophase. At the end of telophase. Because the nuclear membrane disappears at the end of a prophase, and again it reappears. So that is also function of the common function. Now let us discuss the functions of rough endoplasmic reticulum. Functions of rough endoplasmic reticulum. So here, why it is rough? Because ribosomes are present. And what is the function of ribosomes? Protein synthesis. So naturally, the functions will be, that means here the ribosomes on surface synthesize proteins. So that is one of the important functions. Then the second function you can write, that is the rough endoplasmic reticulum bears enzymes for modification of polypeptides. And this process is known as glycosylation. This is one of the very important points from examination point of view. That means the proteins which will be synthesized in the ribosomes, that will enter into the rough endoplasmic reticulum. So that polypeptide will be modified, and that process is known as glycosylation. Then there will be in some to some proteins, oligosaccharides will be attached, and that is known as glycosidation. Then the oligosaccharides are added to proteins to form glycoproteins. And the process is known as glycosidation. It is known as glycosidation. Now this glycosidation is also very important. But this glycosidation starts in endoplasmic reticulum, but completed only in Golgi body. So here let us write starts in area, but completed in Golgi body. This is also very important. Now the next point is that is it provides enzyme precursors. precursors for formation of lysosomes. As already I have told you in the beginning that the endoplasmic reticulum, Golgi bodies, lysosomes are vehicles that form endomembrane system. And inside the lysosomes there are about 50 enzymes. So the precursor of the enzymes, because enzymes are proteins, so the precursor of enzymes are formed by this rough endoplasmic reticulum to which the ribosomes are attached. Then the next point is that it can gives rise to RER, gives rise to SER. Because if ribosomes will be removed, it will be converted to SER. And also another is it provides large surface area, that means 2/3 of the membrane structures are only rough endoplasmic reticulum. So you see these functions are completely separate from the common functions. Now let us discuss the functions by smooth endoplasmic reticulum. So the functions of smooth endoplasmic reticulum. Now from the smooth endoplasmic reticulum, it is very important because the smooth endoplasmic reticulum is responsible for synthesis of all types of lipids, maybe phospholipids, or it may be cholesterol, or it may be steroids, or all types of sex hormones. All these are formed in the smooth endoplasmic reticulum. So that is the most important point. That is a major site for synthesis of lipids, phospholipids, cholesterol, steroid, or you can write steroid hormones, and steroid hormones include sex hormones, and also that is adrenal hormones. This is very, very important from examination point of view. That is synthesis. Then the second important function is the synthesis of ascorbic acid and visual proteins. That means the retinal proteins. Ascorbic acid is vitamin C. Then the third important point is it has a very important role in glycogen metabolism. In liver cells. So glycogen metabolism means both glycogenesis and glycogenolysis. That means both synthesis and breakdown of glycogen inside the liver cells takes place through this smooth endoplasm. That means both glycogenesis and glycogenolysis. That means both breakdown and synthesis take place here. This is also very important. Then the next is detoxification. Detoxification in liver cells. There are certain poisons. The poisons can be detoxified, and particularly you can take the case of barbiturates. They are detoxified by this method. So for this detoxification there are certain enzymes, and the enzymes is usually oxygenase, and there are two cytochromes, that is P450 and P448. So with the help of this oxygenase enzyme and the cytochromes, P450 and 448, usually it carries out the detoxification function. Then the next function is that is uptake and release of calcium ions for relaxation and contraction of muscle cells. So that is why here the smooth endoplasmic reticulum is known as sarcoplasmic reticulum. It is known as sarcoplasmic reticulum. Then another function is there. This smooth endoplasmic reticulum, it gives rise to different types of other organelles, which may be Golgi body, lysosomes, or vacuoles. Gives rise to, that is Golgi body, lysosomes, and vacuoles. So see, the functions are completely different. These are the 7 functions which are carried out by both rough and smooth endoplasmic reticulum, while these 6 are specifically by rough endoplasmic reticulum, and the last 6 functions are specifically carried out by smooth endoplasmic reticulum. So the functional part of this endoplasmic reticulum is very important from a general point of view. So you can take a snapshot and very easily you can remember it. So thank you very much. Wish you a very bright future. God bless you.